Okay, and then we can also look at retroviruses. So retroviruses are the term retro, it's meaning backwards here. And it's because it's going to take RNA and make DNA strands out of it. So typically in our animal cells, we're taking DNA and turning it into RNA. We're not working backwards. And so our cells don't have the kind of equipment to do this. And the retroviruses have to carry around an enzyme that can do that. And so that's reverse transcriptase. This is RNA-dependent DNA polymerase. So it uses RNA as a template. It's dependent on the RNA as a template to synthesize DNA. And so here we have a positive single-strand RNA. It will use its own reverse transcriptase to make a kind of complementary, so the negative single-strand DNA, which it'll then make use the host cells kind of equipment to make the complementary DNA strand, and then from there it will you know, do translation or transcription and translation in a way similar to, what's typically done in the cell. The genome replication is used to make more of the positive single-strand RNA. But what also is important about these retroviruses is when they make the DNA, they're not always going to make a bunch of copies of themselves. They may make double-stranded DNA, and then that can become integrated into the host genome and remain latent within the host genome. Okay, and then assembly, um, similar in that the cells or the virus particles are going to self-assemble within the host cell. There's a few ways for the release to happen. Um, the cells that, or the viruses that have viral envelopes will actually bud off of the host cell. And in doing so, they're coated by the cell membrane, right? So becomes the viral envelope. In doing so, the host is not immediately killed. And so it'll remain kind of an intact cell, and more copies of the virus can bud off. In those animal viruses that do not have a viral envelope, um, they exit a little bit different. So they will induce apoptosis, so cell death, and then the viruses are released from the cell. All right, and viruses change very quickly. And so I'll just talk about this pretty briefly. But there's two kind of names given to the way that the viruses change. One is antigenic drift. And so the major reason here is that replicase, so that enzyme that they're using to make copies, it's doesn't have any kind of proofreading mechanism. So unlike when our animal cells are copying our own DNA, there's some kind of proofreading and making sure everything is all right. With the replicase, that doesn't occur. And so mistakes happen and these mutations can accumulate. Some of them are going to, maybe be negative mutations, but there's going to be enough of them that some of these mutations are going to be helpful in avoiding the immune defenses of their host. In this antigenic drift is why we need new flu vaccines each year. There's enough kind of changes, mutations acquired over the course of the year in the flu viruses, just just due to the kind of mistakes being made over replication. That you end up with different like the locks on the or the spike proteins on the outside of the virus. Another way for viruses to change is through antigenic shift. And so this is essentially mixing of DNA during a multivirus infection. So if a host is infected by multiple virus types at the same time, um, those bits, those bits of RNA that are inside that cell can end up mixing up with each other. And this can cause formation of a new virus subtype. And this we will talk about a little bit further when we talk more about influenza.